Eder, Lilia, Romero, coming from Buenos Aires. Thank you so much for making the long trip.
Well, thank you for this nice invitation. And the thing, okay. And the pointing, this is the point. Okay. Good morning to everyone. So I want to bring you to hear what do you think they have in common, these all different things. Tumor cell, a bacteria, and a parasite. All these things share in common the fact that at a certain point of their life cycle, they display resistance. And that is because of the expression of efflux pumps that make them resistant to the treatments with antibiotics or to chemotherapy. So this is a very important thing in common. So in order to build a therapeutic strategy based on nanoparticles against some of these targets, one of the most important things is to start well. So first we have to know where the target cells are. And depending on this, we have to prepare nanoparticles suitable for different routes of administration. For instance, if the target is in the circulating cells, we have to administer the nanoparticles by the intravenous routes, or in the skin, or internal organs, in the gastrointestinal tract. They all deserve different pathways of administration. And in this case, I will talk about nanoparticles which are going to be used against cell targets which are in the respiratory tract, in the lungs, in the lungs. So the nanoparticles suitable for inhalation and non-invasive route has to be prepared. And once that we have stated this, we have to know where is the target site inside the cells, because it will be different according to each pathological landscape. For instance, in the case of tumor cells, our target can be in the plasma membrane, but in the case of infections, in the case of a resistant bacteria, for instance, the target can be into a phagolysosomal compartment, or it can be, in case of Plasmodium, in a very complex intracellular environment where many parasites are surrounded by parasitophorus vacuola. And in all these cases, we have got our common enemy, which is our target. That's the expression of this multi-drug resistant efflux pump. So what I'm going to tell you right now, it's not exactly a strategy against a parasite, but the general lineaments can be followed if you want to build an effective strategy against an intracellular target, which can be, of course, a parasite. What I'm going to tell you right now is something which is useful in our research to efficiently target this type of intracellular parasite, which is a resistant mycobacterium. If we want to get to this intracellular target, which is the parasite inside phagolysosomal compartment expressing this pump that pumps out these two types of antibiotics and makes it resistant to this treatment, what we have to do is to count on nanoparticles that can bring this one or the two together antibiotics. But the co-delivery of these two factors is not enough. We do need something else, and this is the inclusion of a factor that can block the activity of this pump. I'm talking about an inhibitor of this multi-drug resistant pump, and the co-delivery of all these three factors together can really make a difference when you want to target a parasite of this type. But if the nanoparticle is going to be taken up by an endocytic mechanism, then we have to know that the nanoparticle will also be entrapped into an intracellular vesicle, an acidic vesicle. And if so, the cargo will not be available to target extra endolysosomal targets. So what do we have to do? We have to include an escape mechanism in the nanoparticle to ensure that these drugs can be released and they can achieve an intracellular, intracytoplasmic target. And the same occurs with the inhibitor. And only after that, we can be sure that a huge concentration of intracytoplasmic released antibiotic is achieved. And hopefully, after that, our bacteria will be killed. Well, summarizing, it seems to be that to kill a microbe like that, something very complicated we have to do. Something that seems like a science fiction target that no pharmaceutical company would be willing to fund. However, I will tell you what we have done in Buenos Aires to face problems like this under this statement: the simpler, the better, and use natural products and do not use any chemical derivatization to produce your nanoparticles. And I will briefly speak to you about the Archaeosomes portfolio we have developed with the help of this small startup that was created last year in our university. The Archaeosomes are nanoparticles that contain archaeolipids, and the archaeolipids are extracted from archaebacteria. And because of their origin, which is completely different from the conventional ones, which are the sources for phospholipids used to prepare liposomes, they are not extracted from animals, not from plants, not from microorganisms, not from bacteria, not from fungi. They are not synthetics, they are archaeolipids, and they display several interesting properties that make them quite different to the conventional liposomes. This is a typical archaeolipid, so the structure is quite different to the one from conventional phospholipids, and because of that, they are resistant to hydrolytic, enzymatic, or chemical attacks, so they are not so easy to be destroyed in a biological compartment or in the shelf life. And they also can be extensively taken up by macrophages because they are a natural ligand of a scavenger receptor class A. So we have got here the targeting without any derivatization. And also because of their particular structure, they are particularly low permeability. They have low permeability to protons and hydroxyls. Because of that, they can act as acid or alkaline bags, enabling the dissolution of drugs which are very difficult to be formulated otherwise, without the need of including any artificial gradient of protons. And here there are depicted some other interesting properties of the archaeosomes compared to the liposomes. The archaeosomes, the bilayer, has got a very low rotational and lateral mobility and very low bilayer order, so they are highly entropic, and they act like a kind of glue as compared to the one of liposomes. And because of this, it is very difficult to dissolve any hydrophobic active principle in their inner core. That's characteristic features of the archaeosomes. But there are two more other things I want to point out. Because this type of nanovesicles can be submitted to lyophilization and to heat sterilization, while liposomes do not. And this is very important from the pharmaceutical point of view. The archaeosomes are also very highly taken up as compared to liposomes, independent of their size and origin and composition, by different cell lines, and we have made profit of these interesting properties to prepare different types of nanoparticles that contain archaeolipids and use them in different therapeutic preclinical approaches. And this one, we have got one single nanoparticles with 2 ligands containing this immunomodulator that were used as an adjuvant in the search for a therapeutic vaccine against Chagas disease. And surprisingly, in this search, we found that these acid bags acted as a very efficient immunochemotherapy against Chagas disease because they provided full survival in front to a lethal challenge with Chagas parasites, and also decreased completely the parasitemia in infected mice. These are unpublished results. We also have prepared ultra-small solid lipid nanoparticles, where the nanoparticles are fully covered by these archaeolipids. And we have succinctly used them as very efficient reducers of intestinal inflammation. But back to the endocytic uptake of archaeosomes, as you may know, we do know that they are taken up by different endocytic mechanisms that we have studied, and all of them end up into the same compartment. Most of the cargo ends up in this acidic compartment. Here we can see the archaeosomes inside the lysosomes of macrophage. We also do know something else. We do know that the archaeosomes are early endocytic apoptotic agents, different to liposomes, which are necrotic inducers. But knowing this, as I told you before, we have to include a pH-sensitive mediated escape mechanism if we want that our cargo is released and can target the cell membrane or any other extralysosomal target like this one. So we have managed to prepare, on the basis of the structure of a pH-sensitive nanoliposome, what we have called the pH-sensitive archaeosomes, and we have used them very efficiently to have a very good nanoparticle that could provide a huge intracytoplasmic drug delivery because of the combination of the targeted of the increased endocytic uptake because of the presence of the total polar archaeolipids, and also the pH-sensitive mechanism. So we managed to build this nanoparticle capable of the highest cytoplasmic drug delivery and used it very efficiently to have anti-inflammatory agents. But this is not all. These nanoparticles can also be nebulized because of the presence of the total polar archaeolipids that increases the shelf life and also the resistance to the shear stress of this colloidal nanoparticle. You know that nebulizing liposomes is not so easy. And I'm talking about colloidal nanoparticles, very soft nanoparticles. And our recent findings show that we can nebulize very safely these pH-sensitive archaeosomes because they are harmless for this surfactant monolayer that covers the alveoli before entering to the alveolar macrophages. And we can nebulize these nanoparticles without also improving the functions of the pulmonary surfactant because the surface tensions is in fact decreased. So we are not causing any harm, and also you can say what happens with these soft nanoparticles encounter this barrier because they can be destroyed, and then the targeting to the underlying cell would never occur. Well, the fact is, we have also found it out recently, that the nanoparticles, they can cross this barrier and they can be taken up, conserving their pH sensitivity. So there are still a very huge intracytoplasmic drug delivery. So overall, targeted delivery of nebulized colloidal nanoparticles is possible. And I'm about to finish. On this basis, I will tell you about this proof of concept, which is very new results I'm talking about the curcumin-enhanced pH-sensitive archaeosomes. I know this is incremental research. This is incremental research, but it seems to work. What I'm talking about? I'm talking about the curcumin, which is one of the main ingredients of the curry. And this is a very interesting molecule because it has got a number of interesting pharmacological properties which cannot be readily available because of the low aqueous solubility of this drug. But we have managed to include this curcumin into the structure of the pH-sensitive archaeosomes, and therefore we have got a nanoparticle capable of providing a number of properties such as antioxidant, anti-inflammatory, anti-tumoral, and of course a blocker of the multi-drug resistant pumps. Hopefully, then we have got the three components that I told before: pH sensitivity, a ligand without any chemical derivatization, and a blocker of the efflux pump, and the combination of the two drugs that can be only one of them. As a conclusion, we have got a nanoparticle that can combine in a pH-sensitive targeted structure antibiotics and curcumin that can be nebulized, and also this is a product made with natural lipids which are fully underexploited and extracted from sustainable resources. Thank you very much. That's it.
Thank you very much. That was extremely interesting, but we have to postpone questions to the coffee break, and we have to move on